Hello everybody and welcome to today's talk in our Evolution and Ecology seminar series. Today we are very excited to have with us Dr. Melissah Rowe. Melissah is a tenure track group leader at the Netherlands Institute of Ecology in Wageningen. She did her undergrad at Macquarie University before obtaining her master's degree and her PhD from the University of Chicago, where she studied sexual selection and sperm competition in fairy wrens. After that, she was a postdoc at Arizona State University and then the University of Oslo, where she then got a grant to continue her research as a researcher, looking into the effects of microbes on ejaculates. In 2019, Melissah moved to Wageningen. She combines fieldwork with lab experiment and molecular work to study evolutionary, ecological and microbial impacts on reproductive biology, mostly in passerine birds. The title of today's talk is Reproductive Microbiomes and the Functional Evolution of Avian Ejaculates. As usual, we have a Q&A session directly after the talk with Melissah. So please do post your questions in the designated Slack channel. The link can be found in the description of this video. So yeah, without any further delay, Melissah, thanks again for accepting our invitation, and the floor is all yours.
Thank you. Okay, let's start with just sharing my screen, which you should now be able to see. Fantastic. Okay, so as I was nicely introduced already, I probably don't need to tell you, but my name is Melissah Rowe. I'm joining you from the Department of Animal Ecology at the Netherlands Institute of Ecology. So I am obviously not Dutch, but I'm here struggling to learn the language and homeschool my children during the lockdown in a language I don't speak. So this talk comes as a nice welcome relief. So I want to, first of all, really thank the organisers for a brilliant seminar series. I personally know, and I'm sure this is true for many of other people out there, that listening to this series has sort of really brought some sort of great moments and buoyed our spirits during this crazy pandemic. And I really am just absolutely thrilled and incredibly honoured to be invited and to join a list of what really is a remarkable set of seminar speakers. So thank you very much for having me. So today I'm actually going to sort of divide my talk into two parts. The first part, I'm going to briefly introduce you to reproductive microbiomes, and then I'm really going to focus on my empirical work looking at the functional evolution of avian ejaculates. Just to tell you a little bit about me though, and sort of what drives me, my fascination with ejaculate biology goes back to my undergraduate days, and actually this image. So this was presented to me in class and I learnt about sperm displacement. So for those that don't know, this is a dragonfly or damselfly penis, and it has barbs on the penis that remove sperm from previous matings. And honestly, this just blew my mind. And ever since, I've been fascinated by questions of sexual selection and particularly sperm evolution and ejaculate biology. So those questions have really centered around trying to understand reproductive success in males and why it is so variable. And also focusing heavily on the post-copulatory side, so trying to understand fertilization success. My work has broadly covered sperm biology and ejaculate evolution, and more recently, I'm also really interested in factors that influence sperm biology and ejaculate competitive ability. So factors such as mating behavior or polyandry, also environmental factors such as temperature, and more recently, the impact of microbes on ejaculates. And it's this topic which I want to talk to you about today. So microbes are ubiquitous. They're absolutely everywhere. And macroorganisms like this tree and this deer you see really live and evolve in a microbial world. So it's not really that surprising that these animals and plants host millions of microbes in and on their bodies. Over the last sort of 2 decades, we've experienced what some call the microbiome revolution. So what we've seen is really growing knowledge of host-associated microbiomes, and it's really fundamentally changing our understanding of organismal biology. And it's becoming increasingly well recognized that we need to consider this microbial population in a host to really understand the host itself. Another thing that's really come out of this microbiome revolution is that our perspective has really shifted. We originally really considered microbes as pathogens and disease-causing organisms. What's become really clear now is that the relationship between host and its microbes can be both detrimental and beneficial, but also host neutral. So at the forefront of this microbiome revolution really has been the gut. And what we've found is that the gut microbiome has been implicated in a really wide range of physiological, behavioral functions and effects. Things like development of the digestive system and aiding in digestion. It's been linked to behavioral traits like anxiety and depression, to obesity, brain development, including effects on behavioral traits and cognitive ability, and also to immune system development and immune function. So really, it seems like the gut microbiome is really, really important. It's also been heavily linked to the evolution of hosts. So the gut microbiome, for example, appears to have perhaps promoted the evolution of herbivory across multiple mammalian lineages. So what we know is that plants contain these polysaccharides that mammalian enzymes themselves can't break down. And it's actually the microbial communities in the gut that are responsible for breaking down these polysaccharides. And so therefore these microbiomes are essential for the transition to herbivory. We also see that microbes in the gut have facilitated dietary shifts or specializations to handle foods that have toxic secondary metabolites. And a nice example is the koala, which is specialized to eat eucalyptus. So these sort of adaptive shifts in microbiome are also seen in a number of other systems. And the image here is showing you that they've seen a shift in the microbiome from wild horses, which are eating natural grasslands, to the microbiome of the domestic horse, where it seems that the microbiome has shifted to allow the digestion of anthropogenic food supplies, as well as to deal with other anthropogenic factors. So microbiomes are emerging as these fundamental modulators of host biology, fitness, and evolution. But I really want to point out here that the vast majority of work that has been done on microbiomes is on the gut, the skin, or the oral microbiome. In contrast, we know much less about reproductive microbiomes. And that's what I'm really interested in. I want to understand whether these reproductive microbial communities can impact host biology, fitness, and evolution. So what is it that we know about reproductive microbiomes so far? Well, the fact that they're not very well studied is a little surprising because we have this long-standing knowledge of sexually transmitted diseases, things like chlamydia, gonorrhea, and syphilis. And we have clear evidence that these can impact female reproductive health, often with some serious consequences and disease states like pelvic inflammatory disease, as well as tubal infertility and even morbidity. And in fact, in some cases, chlamydial infections have even been linked to cervical cancers. So we know this all about sexually transmitted diseases, but we haven't gone much further until recently. The other thing that we've kind of known for quite some time is that bacteria can be bad for sperm. What these images are showing you on this side is human sperm. And what happens when bacteria and sperm interact, you can have a situation where the bacteria essentially adhere to the sperm cells, causing the cells to agglutinate. What this then does in terms of the sperm function is it can impact motility, slowing the sperm down, even in some cases preventing sperms from swimming at all. And the other thing that we've seen is that bacteria that adhere to the surface of the cell, sperm cell, can cause damage to the membrane, essentially rupturing plasma membrane of the sperm and causing the cell to die. Now, this image down here shows you a passerine sperm with some bacteria that appear to be attached. And this was something that I discovered very early on, or several years ago, and has really what got started in trying to understand What are they doing there? What are they? What could their effect be? So all of this stuff we've known for a while, but it's been a single microbe approach. More recently, next generation sequencing technologies have really allowed us to explore reproductive microbiomes, and in particular, the female reproductive microbiome. In humans, what we can see is that across different sites in the female reproductive tract, you have distinct and quite different microbial communities at these different sites. One really noticeable effect is that in the vaginal microbiome, we see this massive dominance of lactobacillus. Whereas if further up the reproductive tract, you see a greater amount of diversity in the microbiomes. And what's been really interesting with the studies of female reproductive microbiomes is they've really shifted our perspective. Originally thinking that the female reproductive tract is somewhat a sterile environment, we now see that that's very much not the case. The vaginal microbiome is also dynamic, so it changes over time. What I'm showing you here is just two individuals and their patterns of change in major bacterial taxa. And what I want you to take from these two images is that the taxa change over time in each individual, but those changes can also be different across individuals. And with vaginal microbiome variability, these changes have been linked to a number of factors, including age, self-reported ethnicity, life history phase, menstruation, and pregnancy. If we look a bit more broadly, step away from humans and look to a bunch of other mammalian taxa, we can see that there is some similarities, but also some differences. The pink spots that you'll see here in the human vaginal microbiome are firmicutes, which is those lactobacillus we talked about. And we see that they're also represented in many of the other mammalian taxa. But also we see that the vaginal microbiome of humans is distinct and particularly low diversity relative to those other taxa. So I just want to focus in on primates, both humans and non-human primates for a moment. So what we see is while that human vaginal microbiome is really dominated by lactobacillus, so up to 93% of the microbiome is lactobacilli, in the non-human primates, the relative contribution of lactobacilli is less than 5%. So in this study of these primates, they also looked at diversity of the microbiome, and reflecting the dominance of lactobacillus, we see that humans have a relatively low diverse microbial community relative to these other primate species. What I think was really interesting about this study is that they went on to try and understand what factors might explain this variation in diversity. And what they found was a number of social and sexual factors seem to have an influence on diversity levels across these taxa. There was also a lot of interrelationship between these factors because many of them sort of are different ways of saying the same thing. So I'll just explain briefly. So first of all, they found that sexual swellings, so whether a primate had swelling of the urogenital region, was positively associated with microbiome diversity. Now, interestingly, sexual swelling is also strongly associated with multi-male mating systems. So this leads me to my next step, or next factor, which is promiscuity, or a broad definition of mating system. So what they found is more promiscuous species had a greater diversity of bacteria in the vagina. Social structure was also important, and looking at a more fine-tuned measure of promiscuity, they looked at relative testis mass and also found that relative testis mass was positively related with bacterial diversity in the vaginal microbiome. So interestingly, this result has also been seen in mice. So this is a slightly smaller study. It's just a comparative study of two promiscuous species. So they looked at the California mouse and the deer mouse. And the two really distinct things about these is the California mouse over here in red is both socially and genetically monogamous. In contrast, it lives sympatrically, the deer mouse is socially and genetically promiscuous. And what they found, looking at the diversity of the vaginal microbiome of these two species, they found that the promiscuous species also had a higher diversity of microbiome, vaginal diversity, sorry. And they found this result sort of no matter how they divided up their species or their taxonomic units in this study. The other thing that they found is there's this promiscuous species, so the deer mouse versus the non-promiscuous species, the California mouse, they really show quite distinct ratios of major bacterial phyla. So for example, the difference between Firmicutes in these two species was really obvious looking at the vaginal microbiome. Okay, so we know something about vaginal microbiomes and also other microbiomes of the reproductive tract of females, but what about males? So considerably less is known about males. I've shown you that some microbes could damage sperm, but is that a sort of a chance occurrence or just random pathogens, or do male reproductive systems also have microbiomes? Well, I guess somewhat not surprisingly, they do. So, but the interesting thing with these microbiomes, so over here, if we look at the semen microbiome of males, we see that it's a really different profile to what you see in the human vaginal microbiome. So these are also human studies. So the male reproductive or semen microbiomes are much more diverse than the vaginal microbiome. At the same time, we consider these samples to be what we call low biomass. So even though they're relatively diverse, there's not massive amounts of bacterial cells in there. The other interesting thing with these semen microbiomes is there has been some links to infertility. Basically, we have found some associations between these microbiomes and low sperm concentrations or abnormal sperm morphology. Looking at this a little bit more closely, a recent study that used next generation sequencing found that they could classify samples that had either high quality semen traits like sperm traits against low quality semen traits. They could classify these based on the presence or absence or relative abundance of different bacterial taxa. So what they found was that high quality semen was heavily associated with large amounts of lactobacillus and Gardnerella, whereas the low quality semen was associated with a higher abundance of Prevotella and Bordetella. So what I want to also point out with these male studies, this example I'm giving you here is a nice example of a next generation sequencing approach. But of the 18 or so studies that have been done so far, all but 5 of them have used culture methods. So there really is a lot more to be learned about the microbiome and reproductive systems of males. This important thing also to understand is that mating between males and females has some interesting and important effects on the microbiome. I'm showing you here the results of a study that was done on bedbugs. So these guys are really interesting because they have traumatic insemination, and the male bedbug's genitalia has a distinct microbiome. This study looked at what happened to the microbiomes of males and females when they were mated. And what they found is in the intromittent organ of males, which is this one that is involved in this traumatic insemination, and the copulatory organ of females, they found that relative to the pre-mating state, once they had mated, they really saw these distinct changes in the microbiome profile. The other thing that they noticed is that while before mating, males and females were more distinct in their microbiome profiles, after mating, they tended to converge upon one another. So mating changes the microbiome, and it tends to increase the similarity of the microbiome between the sexes. So why might reproductive microbiomes be important? So I think I've shown you that they can impact reproductive health and fertility. And a nice clear example here is this sperm cell from the male chaffinch, which has attachment of bacterial to its head, potentially resulting in this damage to the cell. But there could be a number of potential evolutionary consequences or repercussions of reproductive microbiomes in males and females. And I just wanted to sort of walk you through a couple of what I think are the important consequences of reproductive microbiomes. Okay. Okay, so let's imagine we have an ejaculate. In that ejaculate, we have some microbes represented by these green bars. These microbes can attach to sperm cells and cause microbially induced damage, reducing the competitive ability of an ejaculate. So this effect is thought to generate selection on males to evolve antibacterial defenses to protect sperm from this damage. And in fact, that's what we see. We see antimicrobial proteins and enzymes present in the ejaculates of males from a wide range of animal taxa. And it's really this net effect between the detrimental side of bacteria and the antibacterial substances which can mediate that damage. It's that net effect which helps to contribute to how well that ejaculate can fertilize over. Now, you could imagine a scenario where this net effect is different for different males. So male A may have particularly poor quality sperm because they're microbially damaged, whereas male B might have high levels of immune defenses or antibacterial activity in their ejaculate, essentially protecting their sperm. When you put those two males into competition, you can see that these microbes in reproductive systems in the males could potentially influence the outcome of sperm competition. So in males, reproductive microbes could have consequences for post-copulatory sexual selection and processes like sperm competition. At the same time, it's important to remember, particularly for internally fertilizing species, that there's a female reproductive tract to consider, and it has its own microbiome as well as its own set of immunological responses. And I want to focus on a couple of sort of interesting interactions that might have some interesting consequences for post-copulatory processes linking to fertilization and paternity success. So first of all, the female reproductive tract has an immunological response to the male ejaculate. And this could be different based on the microbial load of the different males. So the interaction between female immunity and the microbial load of ejaculates can influence how heightened that immune response is. And because sperm can be collateral damage of this immune response, that can have effects on the ability of an ejaculate to fertilize over. And again, if you put males in competition, you might see that there's differential effects on the ejaculate from the female immune response, leading to differences in the outcome of sperm competition. At the same time, we have ecological interactions between the microbes or microbiome of the males and the microbiome of the females. It has its own set of potential effects. It could be that it's more optimal for the microbiome of the male to be more compatible with that of the female. So perhaps if a microbiome in an ejaculate is very similar to the female immune response will be lower, and so therefore there will be less impact on the male ejaculate sperm. Another potential thing that could be going on could be leading to sexual conflict. So here I'm focusing in on the antimicrobial defenses of ejaculates and the female reproductive microbiome. What might be happening in this case is the ejaculate antimicrobial defenses may be beneficial for males because it provides defense against bacterial damage, but at the same time, those proteins might cause disruptions to the female reproductive microbiome, potentially setting up conditions of sexual conflict between the males and females. Okay, what about pre-copulatory sexual selection? So another thought experiment, building on other ideas of parasite-mediated sexual selection. One possibility is that females may be using male ornamental traits, such as this lovely colourful yellow bill of the mallard, and maybe using that information on the reproductive microbiome of the males. Because bacteria passed to the female during copulation can generate significant costs for females, we may expect there to be selection for female choice for avoidance of pathogens, so avoiding mating with particular males. Another alternative, the sort of the flip side of that, is we may actually see female choice for males harboring beneficial pathogen or beneficial sexually transmitted microbes. And a final possibility is that there may be simply female choice for avoiding just high microbial loads. Rather than avoiding specific bacteria or looking for specific bacteria, they might just be avoiding males with high bacterial loads. And I'd like to think that one of my sort of recent, well, not that recent actually, but one of my previous bits of work might sort of point towards this being important. What we found in these male mallards is that males that had a more colourful bill, their semen killed a greater percentage of the bacteria in a functional assay. So males with more colourful bills have better defences against bacterial-induced sperm damage. So this might be a way for them to control microbial load. And so therefore females may be using phenotypic information to avoid mating with males with high microbial loads in their ejaculates. Okay, so I'm making sure I'm okay for time, and then I'm going to move on to the empirical side of this, or my own empirical work of part of the talk. And obviously reproductive microbiomes is a key part of this, but I briefly wanted to introduce the other star of my show, which is the humble house sparrow. So he may be everywhere and very common, but these house sparrows really are great little birds to work with. So a lot of what I will be talking about for the rest of the talk is based on work in house sparrows. Okay, so what I want to try and do today is present you with sort of 4 lines of research that come together to tell a bit of a story, I hope. I want to put an important caveat here is that some of this work is very preliminary, so you're seeing some of these data that are not fully through the analysis process, so some of it's a bit rough and ready, but hopefully you'll bear with me. I want to talk you through these 4 aspects. I want to talk about ejaculate microbiomes, then sperm bacteria interactions, ejaculate immune function, and then turn to looking at seminal fluid protein evolution. And I'm going to start with ejaculate microbiomes. So I mentioned that there's bacteria in ejaculates, and we know that humans have a semen microbiome. But what about wild birds? So looking at the house sparrow, we do indeed find that there is a nice community of bacteria living in your average house sparrow ejaculate. Looking at across about 32 male house sparrows, we found nearly 1,300 different OTUs. And on average, each sample, so each male, harbours about 64 different bacterial OTUs in their ejaculates. And these images over here are just to sort of point out a few of the interesting examples I found. First of all, we found Helicobacter and also Campylobacter, which are 2 potentially very pathogenic groups. And then intriguingly, we also found Borrelia, which is the bacteria that's involved in Lyme disease. So there's a lot of interesting stuff in there, but rather than talking about specific taxa, I wanted to tell you that one of the really take-home findings that I found with this work is that there is incredibly high inter-individual variability in these ejaculate microbiomes. So what I'm showing you here is just a relative abundance of these different bacterial phyla. And what I hope you'll take home from this graph is that across individuals, with each bar being an individual, there really is different ratios of these bacterial phyla in individual males. Sometimes Bacteroides is incredibly dominant, making up 75% of the microbiome. Other times it's way down here at around 10%. So there's a huge amount of inter-individual variability. And this really intrigues me because now I really want to know what causes it, and perhaps more importantly, what is the consequences of this variation. Now, here's where one of my big caveats come in. I'm showing you some data that looks at the ejaculate microbiome of sparrows versus fairy wrens. The big caveat comes in because this work was not designed to be a comparative study, but we're just trying to take advantage of the fact that we had sequenced these two birds for different projects, but all on the same runs. So we wanted to just, hey, what would happen if we looked at them and compared them? And intriguingly, we found that the microbiome profiles of sparrows and fairy wrens were quite different. Again, this is a graph showing the relative abundances of these major phyla. And what I can quickly point out is if you look in the fairy wrens, there's a lot of these actinobacteria. Perhaps a little bit more interesting is the fact that when you look at microbiome composition, we also see this very strong distinction between the two taxa. So sparrows and fairy wrens have very different microbiomes, which is perhaps not surprising. They do live on opposite sides of the earth. They do eat different things. So I don't want to take much from this. But what I found particularly interesting in this comparison is that when we look at the species richness, so a number of OTUs, and the diversity of these microbiomes, we consistently see that fairy wrens have lower species richness and lower diversity of ejaculate microbiomes relative to the sparrows. So why do I find this interesting? Well, I want to remind you that when people have looked at the vaginal microbiome, they find completely the opposite pattern. Now, I know we can't take too much away from these two species, but I find this really intriguing. In the vaginal microbiome, the more promiscuous species have greater richness and greater diversity. Here we see the opposite. And so I think it sort of suggests that we really need to investigate these microbiomes across a number of different species in relation to these social sexual factors in a lot more detail. Okay, so now I want to talk to you a little bit about sperm-bacteria interactions. Hopefully I've convinced you there's bacteria in these ejaculates of these males. What does it do? I'm going to focus on an early question that I asked myself was, are bacteria bad for bird sperm? So what I did is I set up a small experiment where I took from house sparrows, collected a male ejaculate and diluted it into buffer. I then split that sample into 4 equal aliquots. And in 3 of them, I added different strains of E. coli bacteria. And in this case, 2 of these strains were actually E. coli that I had isolated and cultured from house sparrow ejaculate and cloaca. So they're really biologically relevant E. coli strains. Then what I did is I tracked sperm motility over time to see what would happen. And here's where we get some preliminary results. So what I found is, in a nutshell, that bacteria can reduce sperm motile performance in house sparrows. So this is our control sample, so no bacteria. We see that nearly 80% of the sperm are swimming at time 0, so at our start point, and they do slow down over 10 minutes, which is pretty normal for passerine ejaculates, actually. But what I hope you'll see is that when you have bacteria in the mix, that drop in the proportion of motile sperm is much greater. So as I said, this is preliminary data, but I sort of wanted to see if I could give you a bit of a qualitative understanding of what this meant. So what I'm showing you here is 2 videos of sperm motion. This is house sparrow sperm at 0 minutes. This is our control over here, and this is our bacterial treatment over here. I think everyone's swimming along quite happily. After 10 minutes, the situation's a little bit different. So over here is our control. Sperm have definitely stopped swimming, but there's still a decent proportion of them swimming around quite happily. Over here in our bacterial treatment, really have to look for those swimmers, but they're there. But I hope that this will give you some sort of idea that the bacteria in treatments seem to be reducing the proportion of motile sperm and ejaculates. Okay, I'm going to move on again and talk to you a little bit about ejaculate immune function. Hopefully you recall that I said by bacteria damaging sperm, it's going to generate selection on males to defend their sperm. And as I said, we see antibacterial defenses in the ejaculates of a broad range of taxa. But while sperm might be meeting bacteria in their ejaculates, there's also these antimicrobial proteins that are swimming around doing their defensive actions. And this is also true for the birds that I've looked at. So we've been working with some small functional assays of antibacterial capacity. And what we have found is across waterfowl, as well as a range of passerine species, that ejaculates are capable of killing both gram-negative and gram-positive bacteria. We've also been able to hone in a little bit and determine that it's likely to be complement and lysozyme or lysozyme-like activity that is heavily the basis of this antibacterial defenses. So I'm going to focus a little bit on lysozyme for a moment and tell you a bit more about lysozyme. This is an enzyme that is a key component of the innate immune system, and it essentially breaks down bacterial cell walls. It lyses them. So what we have found in house sparrows is, intriguingly, if you compare the plasma concentration of lysozyme to the ejaculate levels, we see that ejaculate lysozyme concentration is both much higher and also much more variable. So I think this is sort of pointing towards it having some sort of role there, and also for there enough variation to be having some sort of impact. And what we also see in these males is that these ejaculate lysozyme levels are positively correlated with the motile performance of sperm. So we see that a male that has more lysozyme in his ejaculate has a greater proportion of sperm that are swimming in vitro assays. Okay, so moving from a single enzyme to sort of ramping up and going to whole seminal fluid proteins. What I have done is also using the house sparrow is used proteomics to try and understand protein evolution in seminal fluid. So I thought I would sort of start off with giving you some information about what a seminal fluid proteome actually is and what we can do with it. So what a seminal fluid proteome is it's essentially an unambiguous identification of genes that encode for proteins that are present in the ejaculates, and they're transferred from males to females during copulation. So it gives us this unambiguous list, and we can then use that list to look at the function of genes that underlie those proteins. So I found that the house sparrow seminal fluid proteome is functionally complex. There's 827 proteins. And interestingly, this proteome was particularly enriched in proteins involved in immunity and antimicrobial defenses, as well as metabolism, sperm maturation, and proteases and protease inhibitors, so proteases are proteins that cut up other proteins. This is quite similar to many other seminal fluid proteomes that have been looked at. So my next step with this work was to use molecular evolutionary analysis to try and understand evolutionary pressures that are acting on protein coding regions specific to proteins that are actually in the seminal fluid. And the first thing that we did is we used whole genome data from 11 different species. And we used an analysis focusing on just the house sparrow lineage to test for rapid evolution of seminal fluid proteins. So we calculated these D/DS ratios with elevated rates of D/DS suggesting rapid evolution. What did we find? Well, at first I thought this was disappointing because what we found is if we compared the seminal fluid to the rest of the genome, we found that the seminal fluid was not evolving more rapidly. But what we did find is some seminal fluid proteins are evolving rapidly, and these seminal fluid proteins were heavily biased towards two groups. The first being immune proteins, or immune genes, specifically things like complement activity, complement C7, C9, Reg4, but also we saw a rapid evolution of sperm egg binding proteins. We then wanted to look across the phylogeny, the 11 species, and look for evidence of positive selection across passerines in seminal fluid genes. And what we found is that the seminal fluid is really an enhanced target of positive selection. So in seminal fluid, nearly 15% of the genes are under positive selection, or there's evidence of positive selection. Comparison, only about 10% of the genome has evidence of positive selection. So there's a lot more positive selection going on in seminal fluid genes than in the genome as a whole. And really what kept coming out and sort of basically hitting me in the face every time I did an analysis was that these genes that were showing evidence of positive selection really were linked to Go terms of immunity. So we saw enrichment in terms and pathways that were linked to immunity in passerine seminal fluid evolution. In particular, things like complement and anti-complement again. And again, these sperm egg interactions or binding proteins kept popping up. Okay, so what I hope I've done today is that I've shown you that ejaculates and house sparrows do harbor bacterial communities and that they have really high levels of inter-individual variation. I've also shown you that some of these bacteria, at least, have significant consequences for sperm motile performance and then potentially fitness also. But hopefully we'll answer that in another study. I've also shown you that avian ejaculates exhibit significant antibacterial activity. And when I look at immune function or seminal fluid proteins more broadly, we really see that the targets of rapid evolution and positive selection in the seminal fluid is heavily dominated by immune genes. So I hope that I've convinced you that microbes or reproductive microbiomes may play a role in the functional evolution of passerine seminal fluid. And with that, I'd really like to thank a lot of people that have been involved in these different studies. And I'd really like to thank everybody for listening today.
Thanks very much, Melissa. Fantastic talk. I'll give people a few minutes or however long they need to start submitting their questions. In the meantime, I might start off with one myself. So the last kind of point that you highlighted again, that a lot of these genes that pop up in seminal fluid are immunity related genes. And to me, maybe we can elaborate a bit more on this. It kind of intuitively makes sense in females to have a lot of immune functions because they're receiving a kind of a foreign ejaculate, right? In males, I'm maybe not so much because the males are interested should be interested in keeping pathogens out of their system as a whole, not necessarily specifically the ejaculate. Or do you think well, I think what you alluded to that as well. It could be that could be interesting that males advertise their lack of pathogens in their ejaculates, and maybe it's something more akin to cooperation between the sexes rather than conflict.
So I'm going to try and tackle a couple of points that you made there. I'm going to start with the last one first. I would say, yes, I did allude to, but I did so with quite a lot of hand waving, I must admit, that this association we saw between the coloration of the mallard bill and the capacity of their ejaculates to kill bacteria. There's a possibility that females might be able to use bill coloration to gain information about that male's ejaculate, either about his sperm quality, because the assumption could be that lots of antibacterials means lots of good quality sperm, or potentially it could be information about lots of antibacterials means there's a low likelihood of a high microbial load. So yes, I can see that that could be more of an interesting sort of cooperation side of things. What I think is really nice to think about with that particular situation is I've often been wondering, lysosome might be good for both of them up to a certain point, right? So this is why I also brought it up as a potential situation of sexual conflict, because you might sort of see some sort of relationship like this, like up to a certain point, lysosome is great for males, good for females as well, or at least neutral for females. But at some point, maybe that lysosome is so great that it can disrupt the female microbiome, because lysosome is fairly, well, it's non-specific, right? Because it's part of the innate immune system. So you can sort of imagine that if lots of lysosome gets into the system, there's some potential for it to sort of wreak havoc on the female microbiome and therefore be detrimental for the female.
Yeah. Thanks. There is a question from the audience now. The question is, has there been work on comparing microbiomes of sperm in hybridizing species? And would you expect that the microbiomes would play a role in speciation?
Okay, that's a good question. So there's been a little bit of work on that in gut microbiomes. Can I forward some slides?
Sure.
Okay, I'm not sure where they are in here, but okay. So my work on the seminal fluid proteome in house sparrows, at the same time I did a seminal fluid proteome on the Spanish sparrow, which is a very, very closely related species. In fact, what's really cool with these two species is that in the past, they hybridized and they formed a homoploid hybrid species known as the Italian sparrow. So they have had extensive hybridization in the past, but currently the levels of hybridization are pretty low. There's not really any good hybrid zones. You can get them to mate and produce offspring in the lab, but out in nature, we don't see that many hybrids. So I sampled some Patrick birds and looked at the seminal fluid proteome. And what I found is that they were, and I haven't got a lot of this to show you here, but what I found is that they were quite different, both in terms of their overall composition. But then even when you looked at, so about 70% of the proteins were shared. When you looked at those proteins, there was quite some differences in the abundance of many of those proteins. And also, what we found is that those sort of phenotypic changes in proteome composition and abundance were linked to genomic selection. So, or measures, we found concurrent indices of selective sweeps on genes that were involved in these, which encoded for proteins that showed these abundance differences. Now, interestingly, complement and anti-complement kept coming up both in the house sparrow lineage and also across passerines more broadly. And again, I'm going to get a little hand wavy, but bear with me. This complement is really interesting for a couple of reasons, right? So this is basically what complement is. So complement is a protein or a complex of proteins that essentially gets together and forms like a little hole punch. And that hole punch goes around and like punches holes in cell walls. Now, what's really interesting is it's a major component of the female immunological response, and complement can go around and punch little holes in sperm cells. But what we do see is all this evidence of positive selection for both complement and complement regulators. So what are complement regulators? complement regulators are little things that basically go around and stop these hole punches from forming. They can be considered like little cloaking devices for sperm, right? Because if you have these complement regulators, then the complement action on sperm is significantly reduced. If you take these complement regulators away, then essentially many more sperm get attacked by these complement proteins and die. So if there's this positive selection going on these complement and complement regulators, I've sort of thought that maybe you could have a situation where you have coevolution between the male and the female of these systems, right? And so essentially in the house sparrow, you have complement and complement regulators. Oops. In the Spanish sparrow, you have essentially a different set of complement and complement regulators. So when a male is mating in their conspecific environment, you essentially have the male is efficiently got his sperm cloaking device on and everything's fine. But when you have a heterospecific mating, you now have this mismatch between complement and complement regulators that could mean that this male's sperm is not cloaked and so is much more exposed to immunological attack from the female. So I've sort of speculated that this could be that these immunity-related proteins might be playing a role in reproductive incompatibilities in these sparrows. Now, unfortunately, I haven't looked at the Italian sparrow in terms of its microbiome, and I haven't looked at F1 hybrids of these species now, but I do have some work that will eventually come out on sperm proteomics of these two species, the stable hybrid, which is the Italian sparrow, and also F1 hybrids that are produced in captivity of the house and Spanish sparrow.
Cool.
So I think, so to answer that question a bit more completely, if microbes are driving the evolution of these immune genes in ejaculates, you could see this sort of microbial-induced trajectories of proteins, which then can cause incompatibilities in heterospecific matings.
Nice, yeah. Okay, loads more questions coming through. First one is, do you think viruses might be playing any interesting roles in the reproductive microbiomes?
Yeah, I think there's definite potential for that. To be honest, I've started with bacteria because I'm not a microbiologist. And so I think that at the moment, bacteria for me is the most tractable system to be working with. But definitely, I think viruses might be important and are definitely worthwhile looking at. Yeah. There's some really nice pollen virus work that is sort of showing that there's fitness effects.
Nice. I'll look out for that. The next question is there a benefit for females to maintain a hostile environment for sperm to encourage selection for fit sperm?
Yeah, I mean, that is certainly something that we think could be going on, right? So females may have very sort of pH specific environments, or they might have their immunological responses might be targeting sperm cells. So yes, there is an idea. It's a very, I think, plausible idea that females might be maintaining a microbiome even, or a proteome in their reproductive tract that might exert some selective pressure on sperm.
So you showed some nice results of what happened when you added bacteria to an ejaculate. Is it possible to use antibiotics to clear the bacteria and then measure sperm traits, or is that not really feasible?
No, well, I sigh because that is like, it's a great idea in some respects. And I'll explain sort of what the challenges are. So first of all, if I gave antibiotics to these birds, I'm having a systemic effect. I'm not targeting the reproductive microbiome per se, okay? So it's then very hard to disentangle whether any of the effects are because of a change in the gut microbiome versus a change in the ejaculate microbiome. So one of the ways I'm sort of hoping to start approaching this now, and so this is a new sort of lot of research that I'm starting here now that I have a position here in the Netherlands. one of the things I'm doing is trying to understand exactly where these ejaculate microbiomes are coming from as well. So it may be that they're coming from higher up in the reproductive tract, from testes and from the ductus deferens. There may be an environmental source component. And the really interesting thing with birds, and part of the reason I think they're so good for these questions, is they have a cloaca, right? And so a cloaca is this common opening, and you have both waste and ejaculates coming out of that common opening. So I feel like there's a very good possibility that you are getting gastrointestinal microbes incorporated into ejaculates. So once we know whether the cloacal microbiome is the same or different to the ejaculate microbiome, we might be able to start thinking about some sorts of antibacterial or antimicrobial treatments. So you could imagine we find that the ejaculate microbiome is just exactly the same as the cloacal microbiome. Then you could think about sort of topical application of antibacterials that might change the microbiome, and that would get away from those more systemic effects that you would have if you just gave something like an oral antibiotic.
Kind of on a related note, I guess, is there something I wondered, is there something, you know, individuals or species that have high reproductive microbiome diversity, are those same individuals, do they have people measured their microbiomes in the, well, in birds there's a direct link, I suppose, but in other animals, is there covariation between how diverse a microbiome is between these different organs?
I'm not aware of any research to that effect. I know that, well, so within a species, there's been some work in chickens, which is showing that there's relatively large overlap between gastrointestinal and oviduct microbiomes, which sort of suggests that you can get migration from gastrointestinal tract up into the reproductive tract. And they've actually even found quite high up in the reproductive tract, right? So not just into the cloaca, but actually going much further up. Across species, if I think of something, I'll write it in the Slack, but right now I can't think of a study that's looked at that.
Fair enough. Yeah, I wasn't aware of anything either, to be honest, but I'm not an expert at all.
If you look at ejaculate microbiomes, like I was sort of trying to get some numbers the other day, and there's 5 studies in humans, a study in mice, a study in pigs, and bed bugs have been fairly well studied, but it's really a limited number of studies that have looked at ejaculate microbiomes. So I think, you know, there's so much room for people to come and do stuff.
That's quite striking, yeah. Okay, another question from the audience. What about other members of microbiomes, such as fungi? The virus is not a fungi. Would you expect them to play a role in your model organism?
It wouldn't surprise me. I unfortunately give the same sort of responses to what I gave before. I think that would be an interesting thing to look at. I'm intrigued by, I've had conversations with people too, where they've opened up eggs which haven't hatched, and they've found fungi inside the eggs. So I'm really intrigued by, I mean, you know, they're essentially coming either from the female or from the male. They're sort of essentially part of the reproductive microbiome in the broader sense. So I think that would be really interesting to look at, and to seeing whether there's some association with like low egg hatchability. But yeah, fungi, all members of the microbiome, right? Not just the bacteria.
Right. Okay, final one maybe. The presented data on microbiome composition between sexes, between species, were structural. What about the functional role of specific taxa?
Yeah, good question. I feel like often when I'm asked questions about reproductive microbiomes that aren't in humans, which is not my area, and aren't in maybe lab mice, which is also not my area, my answer to much of this is, good question, I don't know. We don't really know much about the function at this point. Certainly not in my system. We've been trying to do some shotgun shotgun proteomics and shotgun metagenomics to try and understand if we can get at function. with challenges at this point, so I can't say that we've had massive amounts of success yet. We've also been taking a more computational approach, and that's starting to reveal some interesting things, but not much that I can really say with anything concrete at the moment. Yeah. But I think trying to understand the functional consequences and which aspects of the microbiome are responsible for those functional consequences is a really important next step.
Certainly, you won't run out of questions anytime soon. All right, that's about the end of it in terms of time. Thanks again very much for a really interesting talk and for a great Q&A session. Thank you.
Thank you.
And everybody else watching, next Wednesday, we'll have with us Li Zhao from Rockefeller University in New York. And the title of Li's talk will be The Origin and Evolution of New Genes and Phenotypes. So please join us again next Wednesday, same time. Keep up to date with our schedule by following us on Twitter and or by joining our Slack channel. Thanks again for watching, and goodbye.